In the rice locus question, we tell you that in rice, locus A and B are genetically separated by 20 map units. We tell you that locus A has two alleles, big A, little a, and that locus B also has two alleles, big B, little b. In this specific question, we tell you a plant heterozygous at both loci is in cis configuration. Just as a reminder, cis configuration means that on one chromosome you'll have the normal or wild type alleles, and that on the other chromosome, as you can see here, you'll have the two mutant alleles. In trans configuration, when we're thinking once again about a heterozygous plant, if it's in trans configuration, you will have on one chromosome one copy of the wild type and one copy of the mutant allele for the other gene, whereas on the other chromosome you'll have the same thing but in opposite. So as you can see in this question, we have the cis configuration. In part A, we ask you to determine the fraction of the gametes produced by this heterozygous plant that will be big A, big B. So in A, we're asking you the proportion that will be big A, big B. So to answer this question, we first need to determine the genotype of the gametes produced. And so to do that, we will use the distance, so the physical distance we have on the chromosome between locus A and locus B, and that's given in the question. We tell you in the question that locus A and B are separated by 20 map units. So now that we know this, we need to determine what will the gametes look like. We know that when the cell undergoes meiosis, we will have 4 gametes that are produced when we start with 2N. In this case, what will happen is that we will end up with by having one gamete that is as the parental chromosome, so just like that. And so these will be the non-recombinant gametes. We know that when the cell undergoes meiosis, the parental gametes will be produced, but there's also crossing over, and in crossing over, the 2 chromosomes will exchange genetic material, which will result in giving us 2 other gametes that will be different from the parent one. And so in that case, we would have big A with little b and little a with big B. So I've tried to use colors just so you could follow a little bit. And what we can see here is that these guys are the parental one and these guys are the recombinant one. Now that we know this, and we also know the distance between locus A and B, to determine the proportion that will be the recombinant and the proportion that will be the parental, we only need to use the formula that states that the percentage of recombination is equal to the distance between the genes in map units. And so if we know that the distance is equal to 20 map units, well, we know that the percentage of recombination is also equal to 20. So if we know that, this means that 20% of our gametes will be these 2 types, and the parental, which is the rest, will be 100 minus 20, which is 80%. In question A, we're asking you what portion will be big A, big B, and we know that big A, big B is a parental gamete, and since we have 80% for the parental gametes, each of these gametes will represent 40%. And so in question A, 40% or 0.4 will be the proportion or the portion of gametes that will be big A, big B. In part B, we ask you if this plant undergoes self-fertilization, what fraction of its progeny will be, and we give you 2 different genotypes. So to determine specific genotypes, what we'll have to do is use a Punnett square. But we won't fill out the whole Punnett square, we'll only look at what we're specifically interested in. So to do this kind of Punnett square, what you need to do is write down on one side what are the gametes that will be given by one parent and what are the gametes that are given by the other parents. In this case, since we know that this plant undergoes fertilization, we'll have the same gametes here and here. And we've already determined previously in part A what the 4 gametes are and their respective proportions. So all we got to do is write them down in our Punnett square. Now that we have each respective gametes on each side of our Punnett square, all we got to do is fill out what's specifically interested in our case. So what we're interested in is for part 1 of question B is having AB on one chromosome and little a, little b on the other chromosome. So we know that we will obtain this if we have from the first parent a gamete big A, big B, and from the second one we would have to have little a, little b. In that case, the resulting genotype would be big A, big B, little a, little b. And to determine the proportion, we only have to multiply these two, because we know that to have this genotype, we must have this gamete and this gamete. So 0.4 times 0.4 gives us 0.16. We know that we can also obtain big A, big B, little a, little b genotype by having this gamete from the second parent with this gamete from the first parent. So this would also give us big A, big B, little a, little b. And once again, to obtain the proportion of these two, we'll just multiply 0.4 by 0.4, which gives us 0.16. So we know that there are two possibilities to obtain in part 1. On one chromosome, big A, big B, and on the other chromosome, little a, little b, and that's in these two. And we know that since we can have either this one or this one, we will add these two proportions to know what's the probability of getting the progeny big A, big B, and little a, little b. So this will be 0.16 plus 0.16, which gives us 0.34. In part B, what we have here is no slash. Instead, we only have the given genotype big A, big B, little b. If we don't have a slash here, it means that it can be any combination of these specific alleles. So we don't mind if they're on the same chromosome or not. We just want these alleles to be there. So we know that we have the possibility to have this, because here we see that it's heterozygous. It has big A, big B, little b. And here it also has big A, big B, little b. And we know there's another option, there's another way to get that, is if, for example, you would have big A, little b. If you have big A, little b, what we're missing is a little a and a big B. So that would be this genotype, which is right there. You would have big A, little b, little a, big B. To obtain the proportion for this one, we multiply 0.1 by 0.1, which gives us 0.01. So far we have this one, this one, this one, and we're missing, for example, if we would get this gamete from the first parent, little a, big B, we would need from the other parent, big A, little b, and that's right here. So in this case, we would once again have the genotype, little a, big B, big A, little b. And to obtain the proportion for this one, we only multiply 0.1 by 0.1, which gives us 0.01. And so in all four of these cases, we have the genotype big A, big B, little b. And to obtain the final portion of this progeny, we will add all of them up together and not multiply them, because we can have either this one, or this or that. So we know that it's going to be 0.16 plus 0.16, which gives us 0.32. Sorry about that. And we add these two, which is 0.01 and 0.01, which gives us a total of 0.034.